A pipette, pipet, pipettor or chemical dropper is a laboratory tool commonly used in chemistry, biology and medicine to transport a measured volume of liquid, often as a media dispenser. Pipettes come in several designs for various purposes with differing levels of accuracy and precision, from single-piece glass pipettes to more complex adjustable or electronic pipettes. Many pipette types work by creating a partial vacuum above the liquid holding chamber and selectively releasing this vacuum to draw up and dispense liquid. Measurement accuracy varies greatly depending on the style. History: the first micropipette was patented in 1960 by Dr. Hans Schmitz. The founder of the company Eppendorf, Dr. Heinrich Nethler, inherited the rights and started the commercial production of micropipettes within the 60s. The adjustable micropipette is a Wisconsin invention developed through interactions among several people, primarily inventor Warren Gilson and Henry Lardy, a professor of biochemistry at the University of Wisconsin-Madison. Nomenclature: although specific descriptive names exist for each type of pipette, in practice any type of pipette will merely be referred to as a pipette, and the desired device will be obvious from context. Sometimes, pipettes that dispense between 1 and 1,000 AI1/4 are distinguished as micropipettes, while macropipettes dispense greater volumes. Common pipettes: air displacement micropipettes. Air displacement micropipettes are a type of adjustable micropipette that measured volume between about 0.1 AA microliter to 1,000 AA microliter. These pipettes require disposable tips that come in contact with the fluid. The 4 standard sizes of micropipettes correspond to 4 different disposable tip colors. These pipettes operate by piston-driven air displacement. A vacuum is generated by the vertical travel of a metal or ceramic piston within an airtight sleeve. As the piston moves upward, driven by the depression of the plunger, a vacuum is created in the space left vacant by the piston. The liquid around the tip moves into this vacuum and can then be transported and released as necessary. These pipettes are capable of being very precise and accurate. However, since they rely on air displacement, they are subject to inaccuracies caused by the changing environment, particularly temperature and user technique. For these reasons, this equipment must be carefully maintained and calibrated, and users must be trained to exercise correct and consistent technique. The micropipette was invented and patented in 1960 by Dr. Hans Schmitz of Marburg, Germany. Afterwards, the co-founder of the biotechnology company Eppendorf, Dr. Heinrich Nethler, inherited the rights and initiated the global and general use of micropipettes in labs. In 1972, the adjustable micropipette was invented at the University of Wisconsin-Madison by several people, primarily Warren Gilson and Henry Lardy. Micropipettes brands include Gilson, Ergo-1, Eppendorf, Hamilton, Rainin, Drummond, Brandtech, Oxford, Hirschman, BioHit, Labnet, Nishirayo, Gilson, Corning, Vistalab, Thermo, Jenquans, Vertex, Handipet, and Precis. Several different types of air displacement pipettes exist: adjustable or fixed, volumed handled, single-channel, multi-channel or repeater, conical tips or cylindrical tips, standard or locking, manual or electronic. Manufacturer: irrespective of brand or expensive pipette, every micropipette manufacturer recommends checking the calibration at least every 6 months, if used regularly. Companies in the drug or food industries are required to calibrate their pipettes quarterly. Schools which are conducting chemistry classes can have this process annually. Those studying forensics and research where a great deal of testing is commonplace will perform monthly calibrations. Positive-Displacement Pipette: these are similar to air displacement pipettes, but are less commonly used and are used to avoid contamination and for volatile or viscous substances at small volumes, such as DNA. The major difference is that the disposable tip is a microsyringe, composed of a plunger which directly displaces the liquid. Volumetric pipettes. Volumetric pipettes or bulb pipette allow the user to measure a volume of solution extremely accurately. These pipettes have a large bulb with a long narrow portion above with a single graduation mark as it is calibrated for a single volume. Typical volumes are 10, 25 and 50 milliliters. Volumetric pipettes are commonly used to make laboratory solutions from a base stock as well as prepare solutions for titration. Graduated pipettes. Graduated pipettes are a type of micropipette consisting of a long tube with a series of graduations, as on a graduated cylinder or burette, to indicate different calibrated volumes. They also require a source of vacuum. In the early days of chemistry and biology, the mouth was used. Graduated pipettes commonly come in 5, 10, 25 and 50 milliliters volumes. To avoid accidental ingestion of potentially harmful substances, a variety of propipettors have been developed, both entirely manual and electrically assisted. Originally pipettes were made of soda-lime glass, but currently many are made of borosilicate glass. Disposable graduated pipettes are often made of polystyrene. Graduated pipettes are often graduated in one of two ways, mu, backward or drain-out pipettes have a 0 milliliters mark just above the end of the pipette. A volume is pulled into the pipette, sometimes to the maximum volume, then the needed volume is aliquoted out. Serological, forward or blowout pipettes have no 0 milliliters mark as that corresponds to an empty pipette. The volume needed is pulled into the pipette and then dispensed. Historically, the accuracy of a graduated pipette was not as good as that of a volumetric pipette. However, with improved manufacturing methods, the accuracies listed by the manufacturer can equal volumetric pipettes. Graduated pipettes have plus/slash tolerances that range from 0.6% to 0.4% of the nominal volume when measured at 20 degrees Celsius. Graduated pipettes are manufactured according to ISO specifications for accuracy and the arrangement of the graduations. A-grade pipettes are more accurate than B-grade pipettes. Pasteur pipette. Pasteur pipettes are plastic or glass pipettes used to transfer small amounts of liquids, but are not graduated or calibrated for any particular volume. Pasteur pipettes are also called teat pipettes, droppers, eyedroppers and chemical droppers. Transfer pipettes. Transfer pipettes, also known as barrel pipettes, are similar to Pasteur pipettes but are made from a single piece of plastic and their bulb can serve as the liquid holding chamber. Specialized pipettes, pipetting syringe. Pipetting syringes are handheld devices that combine the functions of volumetric pipettes, graduated pipettes, and burettes. They are calibrated to ISO volumetric A-grade standards. A glass or plastic pipette tube is used with a thumb-operated piston and PTFE seal which slides within the pipette in a positive displacement operation. Such a device can be used on a wide variety of fluids in volumes between 0.5 ml and 25 ml. This arrangement provides improvements in precision, handling safety, reliability, economy, and versatility. No disposable tips or pipetting aids are needed with the pipetting syringe. Slyke Pipette, a graduated pipette commonly used in medical technology with serologic pipettes for volumetric analysis. Invented by Donald Dexter Van Slyke. Ostwald Folland Pipette, a special pipette used in measuring viscous fluids such as whole blood. Common in medical technology laboratory setups together with other pipettes. Invented by Friedrich Wilhelm Ostwald, a Baltic German chemist and later refined by Otto Folland, an American chemist. Glass micropipette. These are used to physically interact with microscopic samples, such as in the procedures of microinjection and patch clamping. Most micropipettes are made of borosilicate, aluminosilicate or quartz with many types and sizes of glass tubing being available. Each of these compositions has unique properties which will determine suitable applications. Glass micropipettes are fabricated in a micropipette puller and are typically used in a micromanipulator. Microfluidic pipette. A recent introduction into the micropipette field integrates the versatility of microfluidics into a freely positionable pipette platform. At the tip of the device a localized flow zone is created, allowing for constant control of the nanolighter environment, directly in front of the pipette. The pipettes are made from polydimethylsiloxane which is formed using reactive injection molding. Interfacing of these pipettes using pneumatics enables multiple solutions to be loaded and switched on demand, with solution exchange times of 100 milliseconds, invented by Aller Einler, currently situated in the Biophysical Technology Lab at Chalmers University of Technology in Sweden. The technology became commercial in 2013, under the name Multifunctional Pipette, with the company Avalance Biotech AB. Extremely low-volume pipettes, a Zeptoliter pipette has been developed at Brookhaven National Laboratory. The pipette is made of a carbon shell, within which is an alloy of gold germanium. The pipette was used to learn about how crystallization takes place. Robots. Pipette robots are capable of manipulating the pipettes as humans would do. Calibration. Pipette recalibration is an important consideration in laboratories using these devices. It is the act of examining or adjusting the quality of being near to the true value of a measuring device by comparison with a standard. Pipette calibration is essential to ensure that the instrument is working according to expectations and as per the defined regimes or work protocols. Pipette calibration is considered to be a complex affair because it includes many elements of calibration procedure and several calibration protocol options as well as makes and models of pipettes to consider. References. External links, helpful hints on the use of a volumetric pipette by Oliver Seeley, The History of the Pipette.